Hello, y'all. Welcome to lesson 134, my DP chemistry curriculum. Today we're going over structure 2.4.6. This is an HL topic. So we're going to be covering condensation polymers that form by the reaction between functional groups in each monomer with the release of a small molecule. This being condensation, we can assume that molecule is going to be water. We will represent the repeating unit of polyamides and polyesters given monomer structures. Something we must know is that all biological macromolecules form by condensation reactions and break down by hydrolysis. So that answers the overarching question here, which are how are natural and biological polymers formed? Well, the answer is just condensation. Condensation reactions. I hinted this in the last video, but a condensation reaction is really just the formation of a polymer, and within that we have our two monomers bond and release water. So when we form a polymer by two monomers bonding and releasing water as a byproduct, that is a condensation reaction. A quick way to just visualize it is if we have two structures, structure A and structure B, and these are our monomers. When we add them together, we do get the bond between A and B, but we also have a product of one molecule of liquid water. Now, we generally actually categorize these. So we categorize condensation polymers by their functional group. So it's helpful to know our functional groups, specifically the functional group formed when they create their bond, so after the condensation reaction. Now, with that, we're going to start with our first one, which is actually a series of esters, or what you might know as polyester. So that polyester, what's generally used in fabric and clothing, is nothing but a uh, repeating subunits containing, oops, containing esters. So the most common polyester is actually the one that you see in most of your clothes is what we call polyethylene, terra or terephthalate, phthalate, PETE. Um, and this is a very common polyester, but the structure is simply just, and I say simply, but it's not a simple structure, it's actually quite complex. We have this C double bond O, which creates one part of that polyester along with a benzene, then our actual ester between two separate subunits, and then we have our carbon, hydrogen, hydrogen, carbon, hydrogen, hydrogen, oxygen bond, N. And so this is the polyester that it's generally making up your clothing. This whole section is going to be our polyethylene. This section being our terephthalate. And then we see right here in the middle our ester. You'll even notice that we have half of an ester here and half of an ester here because as we connect them, they continue to make esters, hence polyester. So the ester is formed by the combination of functional groups on the monomers. And you can now tell what those functional groups are if we're pulling water out. So if we look at our first monomer, um, in this case, I'm just going to use a generic example using R to represent our hydrocarbon chain as we've been doing in our organic chem unit. C double bond O, single bond OH, we have our carboxylic acid. Then on our second one, we have opposite an alcohol group bonded to our R. When we add these two together, the oxygen hydrogen from the carboxylic acid leaves with the hydrogen and makes water. So our product of this reaction, and I'll say R prime just to differentiate, is R C double bond O O R prime. And we have our ester. And we also have water as a product. And that's the formation of esters as a condensation reaction. Now, you'll notice here this is just kind of a closed system. It's not a polymer. So when we make polyesters, so that was just a singular ester, but when we make polyesters, something that we must have is we have to have monomers that can do it on both sides. So we might have monomer 1 or A have two dicarboxylic acids. Oops, OH, OH, and then we can add that to B, which in that case is going to be a diol. So we have two alcohols or two hydroxyls. When we add those together, we still keep our C double bond O, OH on one side, bonded to C double bond O, bonded to O, bonded to C, H, and I'm going to have to move all of this because I ran out of room. Whoops. OH. But we can now see that on one side, we still have our alcohol. On the other side, we still have our carboxylic acid, and AB can then bond with another AB. And we can just continuously make these chains. So for a polyester, we need to be able to have that repeating subunit section. So those are polyesters. Taking a break from those, we're going to move over to polyamides. Now, polyamides, now that we know our functional groups, which let me just first say that these form fibers. So we use them in like string and rope, dental floss, string, rope, dental floss. You might use them in rock climbing or pitching a tent or something, anything where you need strong ropes for very lightweight purposes or just that good combo. We're going to generally use polyamides. These form an amide clearly through condensation. So they're going to be produced in the same way as polyesters, but we can go ahead and take a look at an example of nylon, specifically nylon 6,6, and you'll see where that name comes from momentarily. When we look at nylon 6,6, we start with a carbon double bond O, which you should recognize very similar to our polyester. It's the same way we start polyethylene so that we can continue the polyamide or continue the polymer chain. So carbon double bond O, then we're going to have carbon, 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 carbon. You'll notice I've drawn 6 carbons. Double bond O, single bond N, there's our amide, bonded to carbon, 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 bonded to, once again, the other half of the amide. And so we have this massive chain of carbons broken up by an amide. Now, to save time, I'm not going to draw each of the hydrogens here, but you can assume that on each of these carbons, there is a single bonded hydrogen. I just don't feel like doing it. On a DP exam, if you're asked to draw it, you won't be asked to memorize this at all, but make sure to just draw all your hydrogens. Don't be lazy like me. So here is our amide. Now, if you want to know why it's nylon 6,6, you'll notice that on both sides we have 6 carbons, and that's where it comes from. Now, once again, these are produced in the same way, produced in the same way as polyesters. The only difference is now we have an amine, which will become an amide in it. So if we are going to look at the general formula with R's like we did before, we have our carboxylic acid, and this is not for a polyamide, this is just for the single amide there, OH plus HNHR. We're going to continue to see that this leaves. For the polyamide, the only difference is you have, of course, a C double bond OOH, and that's a C, and you have another amine on the other side of that R. So that is going to be our polyamide. I'll just draw the final product of this being R, C double bond O, single bond N, single bond H, bonded to R plus a water. So that is our condensation reaction for polyamides. Now, the last thing that we're going to go over, and it's still going to take a bit of time, are biological molecules. Now, all biological molecules are also produced via condensation, and we have 3 that we're going to focus on. Polypeptides, polysaccharides, and polynucleotides. Now, polypeptides are just proteins, also referred to as amino acids. Polysaccharides are carbs or carbohydrates and sugars. Sugars are carbs, and polynucleotides are what we refer to when we're talking about DNA and RNA. The Na stands for nucleic acid, and the D and R stand for the sugars within it, deoxyribose or ribose. So starting with our polypeptides. So like I said, these are amino acids. And scientists are quite uncreative because the name literally comes from the condensation reaction of a amine and carboxylic acid. Therefore, amine plus carboxylic acid is amino acid. Now, we do know that these are going to therefore make an amide, just like we saw right before. So when we look at the monomer of an amino acid, we have a pretty cool structure here. We have our central carbon with an R group. That's what differentiates the different amino acids. Bonded to a hydrogen, bonded to a carboxylic acid, bonded to an amine. So you'll notice any two can go ahead and form that polyamide with one another because they just go one end to the other. So when we go ahead and look at our polymer, we'll go ahead and look at two repeating structures so that we have our amide in there. So we'll just cut it off, N, H, C, our R group, H, C, double bond O, single bond nitrogen, bond hydrogen, bond carbon, second R group, H, C, double bond O, and we'll cut it off there. And we have N repeating units with our amide right in the middle. And there's our polymer of an amino acid or our polypeptide. So that's about it for those. If we move on to polysaccharides, and once again, that's going to be a condensation reaction. Polysaccharides are formed by the condensation between two hydroxyl groups or hydroxy groups. Which is odd because we haven't seen two hydroxy groups do it yet. So how does that actually work? Well, if we have a monomer of it, which is actually called a monosaccharide, and we'll take one of the most common examples. In this case, we're going to take glucose, just the simplest sugar that we use for energy. Plants create it through photosynthesis, and we consume it as we go through our metabolism in our mitochondria. So this starts with a six carbon ring, though there is an oxygen replacing it, and we have a CH2 little methanol group off the top. OH, 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 and OH. Now, you'll notice I did put some OHs on the bottom and some on top. That is because there are cis and trans versions because it is a ring structure. So remember that these alcohol groups are locked in their position because of the ring. This is HL, so you do need to understand that those do not belong on other sides of the ring. Now, when we take this and we form our polysaccharide, we actually draw it a little weirdly. Saccharide. So for this diagram, we're going to just draw two of them. So we have our same sort of ring, and I'm actually going to cheat here. I'm just going to copy and paste my ring structure from before. And I'm going to duplicate it again. I know it's a bit of cheating, but you can pause the video and do it yourself. And I'm going to remove the ends off. So when we draw our polysaccharide, we have two OHs, and we only need two hydrogens and one oxygen. So we actually have one oxygen remaining on either side. And we're going to cut it off kind of in the middle of that bond, as we normally do. But the thing to point out here is that instead of forming an ester or an amide, when you have those two hydroxy groups, we actually form an ether. This is technically what we would refer to as a polyether because we formed an ether group. A little weird, the DP doesn't actually require you to know that, but you should be able to recognize the functional groups when presented with them, especially as an HL student. So the last one are polynucleotides. Now, these are the ones you actually need to know the least about. Whoops, nucleo. As I was talking, I forgot how to spell tides. So polynucleotides contain three different groups all working together. So a DNA or RNA molecule contains a sugar, phosphate group. Now, this is the same phosphate that you've probably memorized as one of your many polyatomic ions, and a nitrogenous base. And it has a pretty general structure. All DNA and RNA nucleotides look pretty similar. We start with a five-ring sugar, but much like glucose, one of the axis points has been replaced by oxygen. Then we have that fifth carbon sticking up off the fourth carbon, and that's bonded to our phosphate, generally represented by a circle and a P that represents phosphate. The first carbon is bonded to a nitrogenous base. And actually, I'll just point these out. So this is the sugar, that's the phosphate, and that's a nitrogenous base. This would be bonded to another molecule exactly the same as it. And once again, I'm just going to cheat a little. And so right here, this is our bond formed via condensation. That's about all you really need to know for the DP curriculum. I like to add in a little bit of extra, so I'm just going to go ahead and identify some key areas to this. So this sugar is what differentiates a DNA from an RNA, as well as theoretically there is one difference in the base here. These can also be different, but the sugar is either deoxyribose, or ribose. And that's what differentiates those molecules, as well as the nitrogenous base, which for DNA can be G, C, T, or A, and for RNA, it could be G, C, U, or A, where thymine is replaced by uracil. As long as you're in chemistry, you don't need to know that really, but I think it's a good way to kind of bridge those ideas if you're also taking a biology course or DP Bio. The very last thing I'm going to talk about here is how do we break bonds? Because we use condensation reactions to build those bonds, but to break biological molecules, we use what we call hydrolysis, which is just a very fancy way of saying water, hydro, break, lysis. So again, if you know biology, to lyse is when a cell breaks. So when a cell lyses, it breaks. It usually explodes from too much water. But to break and water give us the term hydrolysis. It's basically the inverse of condensation. So just inverse of condensation. You take water out to build the molecule, you put water in to break the molecule. And that's about it. I hope this helps. I hope you can make those connections to your biology curriculum and have a great day.